So you have the equivalent of 32 tons of super in your soil as phosphorus. Now, the question is, why do we actually need to apply phosphorus if we basically have 32 tons of super already within our soil? Now, by the way, that's to a meter of depth in a relatively low phosphorus soil. So the question is, why do we need to apply it? Why does it cost so much? And why can't we tap into it? So you'll learn all of that today in this video, as well as how to actually manage phosphorus in a regenerative manner without costing ourselves yield. So this video is part of our regenerative nutrition series. If you want to go check out the whole series, you can check it out either here on YouTube in the playlist section, or you can head over to a website where I have this written up in a blog. As it is, it's a pretty long blog, but it has everything we talk about here today on the blog, so you can reference any particular point you want there. So in this video, we'll be going over everything you need to know about phosphorus, including what it is and how its role in the plant, how it is in the soil, and that's complicated, as well as how we're testing for it, and then how to manage it as well. So we have all of this, as well as application rates, and then even the fertilizers we can use, how to improve phosphorus efficiency. So let's get into it. Phosphorus is the mineral in the plant after nitrogen, and unlike nitrogen, it completely comes from the soil. So nitrogen we can grow, and effectively it doesn't really matter what the amount is in our soil because we can grow it. Phosphorus, on the other hand, basically 100% comes from our soil unless we apply it, which can be quite problematic if you're in low phosphorus soils. So it can make up anywhere between 0.05% of the plant up to 0.5% of the plant on the dry matter phase. So it doesn't actually make up that much, but after nitrogen, it's probably the next mineral that determines yield. And the reason for that, well, there's kind of two. The big one that phosphorus plays into is the production of ATP. Now, this is almost like a battery within the plant, and it supplies energy to metabolic activities. So anything that the plant wants to do, it requires ATP to supply the energy for that. The other role of phosphorus within the plant itself is the production of DNA and RNA, so that's quite important, as well as phospholipid bilayers, which makes up the cell wall. So pretty important. In terms of the overall plant, it really drives root development. So when plants are deficient in phosphorus, they tend to be stunted and straggly with small root systems. And this is really, really important, and I can't stress this enough. When plants are deficient, meaning that phosphorus management at the very start of the season is critical to making sure we have a really good root system to then make sure they can explore the soil well, which also means it can find moisture, lower in the soil profile, and also other phosphorus later in the season. So the first 10 days, it's critical that phosphorus is supplied to the crop because of root development. So that's super important. It's also quite important for fruit fill or grain fill during the reproductive stage. So the thing about phosphorus that's super difficult is that there's so many interactions that it has within the soil, which makes understanding it really complicated. I've spent many days researching just for this video so I can communicate that hopefully efficiently. So let's start off with phosphorus is available to the plant as either dihydrogen phosphate or monohydrogen phosphate. Basically, it's a soluble form of the phosphate. Now, phosphate is the phosphorus attached to oxygen. Now, this is only available to the plant in a soluble form in the soil. So, unlike nitrogen, for example, where it can take up ammonium or amino acids, the plant can only take up these two forms, and it has to be within the water, which will be a problem, and we'll talk about that why. Now, I recently came across a study that actually suggests that plants can take up organic forms of phosphorus in the form of DNA. So it can actually take up through the roots and then convert that DNA into phosphate within the plant itself. So that's quite interesting, and I think that study was from 2010, so I'm not too sure what the research is up to the uptake of organic phosphorus in the plant, but it's not that widely talked about, and I'm not too sure the extent of the phosphorus supplied to the plant from organic phosphorus. Now, I'd suspect that plants can actually take up phosphorus from the phospholipid bilayer and DNA and ATP directly from microbes in the rhizophagy cycle. So that's something to consider, but typically the majority of our phosphorus supply to our plant will be from phosphates in the water solution. Now, the actual amount of phosphate in our water, in the soil solution here, is typically quite low. So it can typically be about 4 parts per million. So that's not much at all. And the reason why it's so low is that phosphorus here binds rapidly, like really quickly, to the minerals within our soil. So typically there's 4 areas that phosphorus can be in the soil. It can be either in the solution or the water within our soil. It can be in the organic forms. So that's either within microbial biomass, so actually within the microbes. It can be in litter or residue from, say, plant roots. It can be then in broken up components or in soil organic matter, all these things. So there's organic, soluble, sorbed or absorbed phosphorus. Now, this is, in terms of the accessibility, this is super easy. This needs to be mineralized, so it depends on microbial activity. Sorbed is the next easiest way to get phosphorus for the plant. So similar to cations on a colloidal clay, the phosphorus just sticks to or is attracted to positively charged sites within the soil. So that's things like on clay, as well as on iron and aluminium oxides. Now there isn't a chemical bonding between them, they're just attracted to each other, which means they can be taken off more or less in an easy way, but it's still attached there. Now because of that, phosphorus actually doesn't really move that much in the soil. So unlike nitrate, which once it's in the water solution, it's not really going to stick to anything and it'll rapidly just leach away, phosphorus sticks to the soil particles or absorbed onto the iron and aluminium, and it tends to stay there. So it's not going to really move anywhere in the soil, which means we're not going to lose it in terms of leaching. But because of that, we can lose phosphorus with erosion. So if we're losing a lot of topsoil, we're actually losing a lot of phosphorus. So we've got this, and then finally we have our mineral phosphorus. Now the mineral phosphorus is not quite available to the plant at all. A little bit is, but it's super difficult. Now this is stored in the actual parent material of the plant. Typically it's in a mineral called apatite, and then it can also be found bonded to calcium, aluminium, and iron. Now because these are actually chemically bonded, they're quite difficult for the plant or even microbes to break apart, and it takes a lot of effort. So this component of phosphorus is very difficult to access. This component is more accessible and would be accessible even within the season. Organic depends on microbes, and then soluble, quite accessible to the plant. So when a plant takes up, say, a phosphate, it comes out of solution, and the soil will want to maintain an equilibrium between what's in the soil solution and what's absorbed, and to some extent on the mineral phosphorus. So when this phosphorus is taken out of the solution, bits of phosphorus then flow into our soil solution. So there's always almost a recharging into the soil solution. The problem is this process might be slower than what the plant actually requires it to be for it to meet its phosphorus uptake. Now some soils have a greater ability to absorb the phosphorus onto the soil particles. So when you have a lot of iron, so in ferrosols, so you can see a photo here, that really bright red color. So if you're seeing that in chromosols, dermasols, a lot of clay content, those type of soils can absorb phosphorus to a greater extent than, say, light sandy soils that don't have that much clay or iron content, which means less of the phosphorus is going to be in that soluble form and more of it is going to be in the stored phosphorus. Now that can be a bit of a challenge, especially when we're applying phosphorus, because when we apply phosphorus, typically the fertilizers that we apply are in a soluble form. So say we apply our super or a MAP or DAP, whatever, we apply it onto our soil, and then that will, when it rains or it gets wet, it will solubilize into this water component. So a lot of that phosphorus will be then in here, and then after about 6 weeks, this phosphorus will then lock up into this component and effectively become unavailable to the plant immediately. And then it'll be a slower release. The point is, different soils will take that phosphorus fertilizer and store it at different rates. Now you can figure out what more or less that rate is using the phosphorus buffering index. And we'll talk about that in a moment when we talk about testing phosphorus. So typically when we do apply phosphorus fertilizer, only 5 to 20% of the phosphorus actually goes into the plant. So when we think about putting on all this fertilizer, only 5 to 20% of that actually goes into the plant. The rest will go into the phosphorus storage. Now, on the other hand, about 30 to 80% of the phosphorus that the plant takes up is actually from the mineral component of the plant. So not that much fertilizer actually goes into the plant. So whatever the difference is between the uptake of phosphorus will go into our mineral storage. And then the rest of what the plant needs will typically be taken up from the absorbed phosphorus. and then the mineral component will slowly release into absorbed. So sometimes we can talk about losing phosphorus in this system where only 5 to 20% is taken up, but that's not always the case, because next season, potentially that 80% that wasn't taken up will then be that 80% that supplies to the plant. So then if you're doing this basically every year, it works out that last year's phosphorus or previous year's phosphorus in the past will then supply the phosphorus today. Now, another component is that the organic phosphorus can supply, I think in one set it was up to 40 or 50%. So this can actually be quite a significant component of the phosphorus supplied to our plant. So a really important thing to understand with phosphorus absorption is that a lot of it's actually regulated by pH. And at different pH ranges, the amount of calcium, aluminium, and iron absorption will be, or bonding, will be different. So you can see here in this graph, when your soil is highly acidic, so say less than 4%, 4 pH, iron will be the predominant bonding force to our phosphorus. And you can see that that's quite high. So once you get past that pH at 4, a lot of that phosphorus is going to lock up with the iron. Between, say, 6 and 4 is going to be aluminium. Now, there's a little gap here around 4 where phosphorus then increases its availability as these two things kind of drop off. But typically, we're going to assume that's basically all locked away. Once we get between 6 and 7, this is almost like the Goldilocks zone, where we got maximum phosphorus availability. So that's quite good. And then past that 7, phosphorus then starts to bind up with calcium. So one big thing that we can consider when looking at phosphorus availability is our pH. We want to make sure our pH is in that 6.5 zone. If it's too far outside of that, phosphorus can lock up, which means sometimes we don't need to apply phosphorus. We just need to lime our soils or get that pH up. And so lime is a lot cheaper than phosphorus. And so we can make that liming application and try and increase our phosphorus that way. And so when the soil is acidic, dihydrogen phosphate will be the predominant form of phosphorus. And then when it's alkaline or basic, hydrogen phosphate will be the predominant form. I don't think it really matters too much which form it is. So now let's talk about how we can actually test this, because this is super complicated and different tests will extract different components of this to different extents, as well as its ability in different pH ranges. So it's quite complicated in terms of the testing and what test to choose. So unlike, say, a lot of our trace minerals or our nitrogen, there's typically 1 or 2 tests you can pick from. This is a whole range of different tests, different advantages in different soil types and pH ranges. So typically what we have is we can divide them between acidic tests and basic tests in terms of what they can be utilized in. You'll see here the extraction techniques, but what's most important is that if you're in an acid soil or acidic soil, typically we like to use a Bray 1 and Bray 2. So the Bray 1 is what the plant really has available to it more or less immediately. So what we want to make sure is that when the plant is planted or sown, it has available to itself a good supply of phosphorus, which is including this soluble form of phosphorus, as well as some of the absorbed, because the plant can take that by releasing exudates to release that phosphorus. So we want to know what's available to the plant for it to take up in the short term. And that's what we use the Bray 1 for. So this will tell us what is plant available. What we also want to know is what could be available during the season, which is what our Bray 2 looks at. So that's also the exchangeable phosphorus that's likely to be exchanged within that season. So for our basic soils, we have our Olsen for plant available, and then we have our Cowell for plant available plus exchangeable. Now you can also use the Cowell for acidic soils as well, and Cowell is best paired with the phosphorus buffering index to know what the critical value is. So again, that phosphorus buffering index it looks at the ability for the soil to lock up that phosphorus. So it takes it from that soluble component into the stored component. Now I mentioned the critical value. Typically what happens is that when they're developing these tests and determining what application rates or what's the ideal level of phosphorus in our soil, they'll do a bunch of field work. They'll look at different crops and they'll look at the different phosphorus levels, and then they'll determine what is the phosphorus that will determine 90 or 95% of the yield, which is typically what these numbers are. So we're not so much looking at these numbers in terms of what we'd look at the nitrogen numbers, which is the plant needs this much, we need to make sure there's this much in the soil and apply that during the season. What we're looking at here is, yes, we need to make sure that the crop actually has a certain amount of phosphorus in the soil, but we're looking at more or less a certain concentration to allow the plants to pick up that phosphorus without too much effort. And so typically there's a critical value associated with that. So the critical value for our Bray 1 in a typical loamy soil, so not too heavy, not too light, just a nice loamy soil, is about 24 parts per million. Now this will change depending on your clay content. So if you're using our free soil test calculator, which you can get on our website, it's if you have a soil test, you can put all your information and you get a free report label like this with nutritional recommendations. The ideal range or the ideal numbers will change depending on your CEC, which is a function of clay content. And so the higher the clay content, the more phosphorus you want in your soil because it locks up. And so the available phosphorus or the critical range or value for your phosphorus will increase with the increase in clay content and counter exchange capacity. There's also a few nuances with that with the reactive iron and aluminium species, as well as too much calcium in your soil. But this is pretty good. Likewise with our Bray 2, it's going to be higher because it's looking at more of those exchangeable nutrients, so a bigger portion of that pool. And typically we're looking at 48 for a loam soil, parts per million. Now if it was basic, the Olsen will be looking at around 20 and the Colwell about 45. So you can see here the numbers are more or less similar. There'll be a bit of a difference, but really that's kind of what you're aiming for. Now what we like to do in our consulting is also take a total phosphorus test as well. So this looks at not just what is available to the plant, but it looks at all of this. So it looks at what the plant can take up right now, what it could take up in the season, as well as all the phosphorus in that soil, minus the phosphorus silicates, which are basically impossible to break up. There's no point looking at them. So our total phosphorus looks at what could the plant theoretically achieve maybe within 100 years or so. And this is a really, really important number that not many people are getting their soil tests. And we want to know this because typically if you've been farming for a while and you've been putting a lot of fertilizer on, you might have a huge amount of phosphorus in your soil. So we've seen soils with over 1,000 parts per million of phosphorus in the topsoil, which is really pushing up towards that higher end. So with that, it allows us to make a really good informed decision about our phosphorus applications, because if we've got all this phosphorus in our topsoil, there's no need to apply it. But if we don't have any phosphorus in the parent material, because there's simply just not that much phosphorus in the parent material, then we need to make those applications. And so this is kind of how we use it. We have our total phosphorus here, so the big number that represents all of our phosphorus. So this is from an example data. It might have 216. Our Bray 2 will have 21 parts per million. You can see here's stepped down every time because the extraction techniques becomes weaker and weaker. And then our Bray 1 at 14. So this here, and say we're looking at a loam soil here, this tells me it's quite deficient, so we need to be applying a bit more. Likewise with what could be exchangeable, so there's not that much that could be exchangeable. That needs to be up at the 48. And when we take a big step back and go, why are these things not exchangeable? It's simply because there's not that much phosphorus in the total component of the soil. And so that could be that the soil's just old, and older soils tend to have less amount of phosphorus, whereas newer soils tend to have more. Or it could just be that the parent material simply just didn't have phosphorus in it in the first place. And so this just lets us make an informed decision about, do we need to apply phosphorus or not? So in a different situation, if our total phosphorus was at 1,000 parts per million and our Bray 1 was at 14, the question we need to ask ourselves now is, why isn't this phosphorus converting into available phosphorus? And that's likely due to a lack of microbial activity. The question is very much different to, okay, we need to get this number up, to let's convert that over. Typically what we're looking at is about 10% of this total phosphorus being typically what we're looking at is 10% of the total phosphorus being available to the crop. The higher that conversion number, the better your microbial activity. It can be difficult to compare between soils, and typically better within your own soils because of that phosphorus buffering index. But the more microbes you have to cycle and release phosphorus, the more available phosphorus you'll have. Now you can also get another test, which is a water extractable phosphorus, which is this here. That's pretty good. That's important. We have it on our soil tests. Not super important. but it can be interesting to see kind of this step down approach. And then finally, what we've been using more of is the Haney soil test. Now, the Haney soil test, depending on your soil, typically we'll see around 30 for the ideal level, which kind of sits between our Bray 1 and 2 or Olsen and Colwell. What I like about the Haney soil test is that it uses organic acids as the extraction technique, which is similar to what the plant actually uses and the microbes use to solubilize and release phosphorus. So the Haney test will typically do a better job in terms of what is available to the plant. As a American study, I think they've compared 200 or so different soils from across the state, from across the country, and found that the Haney test outperformed another test. So this has been really interesting, and we'll get more data about how this compares to phosphorus uptake in a differential sap test analysis. But I do think it's a really interesting test and worthwhile considering. And again, if you do have a Haney soil test, typically they don't come with interpretation. What we have on our website, again, is our free Haney soil test analysis tool. So you can actually go to our website, put in all your data, and it will return a beautifully formatted soil test with recommendations for you, with a bit of guidance there. Now, before we get into how do we manage phosphorus in a regenerative sense, I think it's quite important to know where the conventional understanding is with phosphorus management. And I don't think it's wrong. I think there's just components of it that lacks a biological understanding. And so it's really important to come from a space where we understand both components. So typically, to work out your application rates for phosphorus, we use a build up and maintain calculation or strategy. So that is, if our phosphorus is low, we want to build it up to that critical value and then maintain it. And so every time you move product off your farm, you're taking the minerals in that product away. And so that, regardless of what it is, whether it's wheat or barley or hay, straw, cattle, you're moving the minerals within those products off farm. Now, for this strategy, the idea is we want to maintain or replace those minerals that we're taking off farm. And so you'll see here the crop removal rates of phosphorus for different crops. Wheat, I think it's about 3 to 3.5 kilos of phosphorus per tonne of wheat. And sometimes when you have excess phosphorus, the plant will take up luxury amounts and then store it in the grain. So there's a bit of a spread on that. It doesn't necessarily mean a quality component, not like protein, but it's important to keep in mind that there is a difference. And so sometimes if there's a massive difference in your phosphorus uptake or removal rate, you can work out the removal rate based on measured data from your paddock. From here, sometimes it's easier to go, what's the average removal rate? But anyways, typically what we want to do, there's two calculations. There's our build up or our P capital, our phosphorus capital, and our maintenance rate. So our maintenance rate will be replacing what the crop removed. So our crop removal is equal to the removal rate times by our yield target. So typically we'll work all this out before putting on our starter fertilizer or before the season and go, how much phosphorus are we going to apply this season? So we're going to estimate our yield target. If you don't know how to do that or you don't have one, we have a video on that, how to estimate it. You can either use rainfall data, which I highly recommend because it kind of steps back from your own operation and goes, what could we achieve as well? Or you could also use historic data. So say for example, our removal rate is 3.5 kilos of phosphorus per tonne, and we're going to aim for a 4-tonne crop. It means that the phosphorus within the grain that we're moving off farm is about 14 kilos per hectare of phosphorus. So at the very least, we want to make sure we can supply 14 kilos of phosphorus for the actual grain to form and the plant to develop, because that's what the plant's going to take away. Now on the other side of this is the build-up component, which contains our phosphorus capital. So this is the phosphorus that we want to add to hit our critical values. So for that, we take the ideal level in our soil test, we minus the current phosphorus levels in our soil test, and then we get basically what we need to add in parts per million. So for example, if we're looking at this soil here, so a Bray 1, and we're going to compare it to that 24. 24 is the ideal. We're going to subtract 14 to that, and it end up with 10 parts per million that we need to supply in phosphorus across the paddock. So we convert that to kilos of phosphorus per hectare and using bulk density of 1.4 to a depth of 10, and we end up with supplying 14 kilos of phosphorus per hectare. So now we're going to add these two together and we get 28 kilos of phosphorus per hectare that we need to add to our paddock in order to bulk up some of the phosphorus supply and supply our crop. So this is going to equal about 140 kilos of MAP fertilizer, which means you'd apply that typically as a starter fertilizer. But if you've been paying attention, you might go, hang on till, if we apply phosphorus, isn't some of that going to then go into our sorbs or mineral component? And the answer is yes. So typically what happens is exactly that. When you apply your phosphorus to say MAP, the MAP will go into this component. It will then, some of that will be fixed. So if we're trying to increase our Bray 1 or Bray 2 to the ideal amount, we're going to need to apply even more because a lot of that's going to get locked up and go into effectively our total. So what we can do here in this calculation here, we work out what we need to apply, but then we're going to apply, I think it's a phosphorus lockup factor or something, but typically we'll be applying 2 to 4 times the amount of phosphorus in order to change our Bray 1 by 1, likewise with any of the others. So we can times this number by somewhere between 2 and 4, so let's just say 3 for example, and we can get that number from phosphorus buffering index when using the Colwell. But let's say we need to apply 3 times as much in order to increase the Bray 1 number by 1. It means that we need to apply 42 kilos of phosphorus in order to get that 14 kilo increase in the Bray 1, because 2/3 of that is going to go into our total. So it's definitely worth the consideration. What I like to do is do that across multiple years. So you want to apply that 42 kilos of phosphorus in your P capital in that 1 year. So you can either do this calculation once and then go on a bit of a program to do that, or what I would rather do is take soil tests every year and then make little adjustments using this to slowly build up your phosphorus, especially in your topsoil. Because ideally for our clients, we're going to be converting a lot more of this phosphorus into available phosphorus, which then should increase that. So I'm not too sure exactly what that lockup factor will be for your soil. So I'd rather just take phosphorus test every year and see how that goes. Now, there's 2 other things I want you to consider. The first one is if you have your ideal level minus your current level, and that comes out as a negative number, it means that you can effectively mine the soil in the top component to bring it down to that critical value and save some money. Now, this is quite useful in 2 situations: when you just don't want to spend the money on fertilizer and the budget's a bit tight, or when the fertilizer is really expensive and you're effectively betting that phosphorus prices will come down in the future. So let's just say, for example, your ideal is 20, and then you currently have 30 in your soil, and so the surplus is 6. That's effectively going to be about 8 kilos of phosphorus per hectare. You can take that 8 kilos and subtract it to your replacement value. So then you'd only need to apply 6 kilos per hectare of phosphorus. Now, some research from the GRDC suggests that if you're going to do this, don't apply any less than 5 kilos per hectare with your starter, because we want to make sure we get a good amount of soluble phosphorus for good root development, because it's so critical that we get good root development so that we can explore more areas in our soil, so that we can take advantage of more phosphorus as well as more nutrition as well as water. So that's a massive part. I wouldn't skimp out on your starter, even if you're regenerative starting off, or ever, I don't think. You can definitely reduce your rates in time, but you want to do this in a very slow manner because you don't want to cost yourself yield. So as I was saying, yeah, you can factor this in when you have excess phosphorus in your soil, but also you can start to almost budget and invest into your soil when fertilizer prices are low or high potentially. So unlike nitrogen, where you're going to lose it anyways within a year, so you want to make sure you're just applying enough. If the price of fertilizer is really low and you're expecting it to go higher, potentially you can increase your capital, phosphorus capital investment to be a bit higher this year, because you know the price is going to be a bit higher next year, so then you can draw on that. So that's some potential strategies in terms of your fertilizer application. So what would you do if you have a heap amount of phosphorus in your total phosphorus and you're trying to move that into your Bray 1 or whatever your planned available measurement is, but you don't have that much? I'll take a combination of approaches. I'll keep your crop requirement amount just so you have that starter phosphorus to make sure you get really good root development. But then potentially I'd reduce the capital P and invest and put that money rather than into P, into say a soil primer or a cover crop to try and increase the biological activity within your soil to then convert that into your Bray 1. And then I'll be using sap tests. We'll talk about that in a moment. To really guide what's happening in this season to then help with next season. So now we're going to go on to the fertilizers we can use, how to make them more efficient, and then a regenerative way to manage phosphorus within our soil. So now that we understand phosphorus in terms of its importance in the soil and all those interactions, and more or less how to calculate it from a conventional point of view, now let's talk about what can we actually do to apply phosphorus to our soil, and then we'll talk about kind of the aims of what we do in our consulting programs, and then how we're going to manage that in a regenerative way. So regarding fertilizers and inputs, we have two types or two categories that I like to put things into: fast release and slow release. So our fast release are typically our inorganic fertilizers. So we have a whole bunch here. What we have is ammonium polyphosphate, which can typically be used as a liquid. I think it's also granular as well. 24%, not bad. Bit of ammonium in there too. So it can supply ammonium. And then we have monopotassium phosphate, 22.5%. Also, you can apply that as a foliar. We have MAP, so monoammonium phosphate, 22%, DAP, diammonium phosphate. Then we've got super at 8.8%, double super and triple super at 17.5 and 20.5. Now the big thing about these is that they release fast. They release or they solubilize, so they release their phosphate components into the soil solution. Now that's great because it's a rapid release of phosphorus to the plant. The problem with that is two things. It locks up after 6 weeks, so the plant has basically all this phosphorus available to itself for 6 weeks, and then it dries up because it goes into the absorbed phosphorus component as well as the mineral phosphorus component as it locks up or it gets fixed. Now typically that's good enough for it to do its job, because really when we're thinking about phosphorus as a starter, you just really want to get it in there, allow the plant to develop a really strong root system, and then the plant will find more phosphorus in the soil as it develops its roots and continues. But from our point of view, where we want to get the mycorrhizal fungi collination happening, which we'll talk about in a moment, this system, we're applying full rate of this, will suppress your mycorrhizal fungi. Because what's the point in the plant giving mycorrhizal fungi all this carbon if it can just take up phosphorus anyways, or without it? So applying these can be a challenge if we want to try and colonize mycorrhizal fungi. So typically, very high analysis, very soluble. It's a good tool. It's a good tool. Next, we have our slow. So these are more organic forms of phosphorus. They typically attach to a carbon component, or they have that carbon component to it, or they're in a bit more of a softer mineral component. So guano, which is basically bat or seabird poo, this is a really good option, about 10 to 12% phosphorus. There's a whole range of other minerals. So a lot of calcium, I think it's 30 or so percent calcium. So you're getting a bit of that calcium in, as well as all the other trace minerals. It's a good overall blend of everything. So that's why that's good, and it'll slow release to the plant over that time. So that's beneficial because it goes into that organic component of the phosphorus reserve, and then biology, and the plant will slowly access that. So if you want to see the chemical breakdown of guano, you can see it here. You can see it has everything. Some people claim it has 60 plus minerals, of which probably most of them are used by the plant that we don't know about, but at least it has the main things that we're looking for. Soft rock phosphate is one of the mineral ones. It's a soft clay that didn't reach the grade for the rock phosphate that make, say, super or MAP or whatever out of. This is quite good because it's also a slow release. That's about 7 to 10%. We also have reactive rock phosphate. The difference between these two is that reactive rock phosphate basically has a faster reaction than soft rock phosphate, and so just the reactivity is different. So I think reactive rock phosphate will dissolve or release a bit faster than soft rock. More or less similar things. They're mined a bit differently, but the benefit of these is that they also have a broad spectrum of minerals in them, mainly calcium and silica. So silica is really good. It helps the plant tolerate a lot of abiotic and biotic stress factors, so like salinity, too much sodium, a few pest and disease pressures. It's a really important mineral that technically isn't an essential mineral, but it's very, very beneficial. So getting a bit of soft rock phosphate on your paddocks as a source of phosphorus is quite a good idea because it has the calcium and the phosphorus and the silica components too. A really powerful thing that you can do, especially if you have a acidic soil or low pH, is instead of liming, you apply your soft rock phosphate because it has 1/5 the neutralizing value of, say, lime. So you can add this into your liming application to get that phosphorus as well as that neutralizing effect, which will then further increase your phosphorus in your soil. So that's a good option. And then lastly, we've got our manures. So we've got chicken manure, pig manure, cow manure. They're kind of the main ones. GRDC did a study and they averaged a few different manures. They found that pig manure had the highest phosphorus content of 2.4%, then chicken manure at 1.6%, and then cow manure or cattle manure at 0.7%. Good sources if you can get it cheap, and transporting costs can be quite expensive. So if you've got a pig farm or chicken sheds or a feedlot near your farm, then that's a really good source. Likewise, it has the carbon component. I think it might be a bit more soluble or available than some of the other forms. So it can really depend on what you're trying to chase. So a lot of these can be applied as a solid or a granule product, or can also be mixed and dissolved into water as a foliar spray. Now, foliar sprays for phosphorus is not that effective. It can be effective on young growth, but really the phosphorus is required in the roots and is also processed in the roots. So for good root development, we really want it to be supplied through the soil. So you can fix your phosphorus deficiencies with a foliar application, only really on young vegetation, but also doesn't really help too much, or not that I've seen in the literature. There are a few references to phosphorus and disease suppression, especially in fungal diseases, and so potentially foliar applications of that could help, but in terms of yield and driving yield, it's best to supply it through the soil rather than a foliar application. Now, typically we can get funneled down into saying, you know, I'm only using MAP, I'm only using guano, but I think a really powerful thing is using a combination of the two. So adding maybe a bit of MAP to chicken manure, or MAP to guano, or MAP to soft rock phosphate, or whatever, can give you that blend between fast release, which is not always bad, it's good because we want the plant to get up and go, and slow release, as well as combining it with a carbon source. And so when we're thinking about our phosphorus applications, I think the big component we want to really think about is getting it in as a starter. When you put it as a starter, so just below the seed or next to the seed, you provide the seed with that phosphorus to really get its root system going and that initial vegetative growth. That's super important because if we don't have that, it's going to stunt the plant later in the season and we get so many problems down the track. So really we want to develop a really nice root system and canopy so that we can start capturing that light and putting it into our soil to feed our biology. So I would highly recommend for any system to make sure you have that starter fertilizer, regardless of the form. The question is, what are we going to apply it as? How much are we going to apply it as? And what additions are we going to apply it with? But anyways, we want to make sure we get that starter fertilizer in so that we can develop that nice big root system and great leaves. So you can see a photo here of a trial done between a phosphorus deficient crop and not deficient. Massive difference. So it's super important you get that in, regardless of whether or not you call yourself regenerative or conventional. It's not regenerative to not produce a crop, because otherwise you're using up all these resources to not produce anything. So we need to produce stuff, and it's super important you get phosphorus in your starter. Cool. Okay, so let's talk about how do we actually do this in a regenerative way. So when I talk about doing it in a regenerative way, it's more or less talking about how can we do this by using biology to increase the efficiencies of our system while maintaining yield and quality. And also want to do it in a profitable way where I'm not adding all these inputs or additional inputs simply to replace our previous inputs. The overall aim is that we want to apply just enough phosphorus for root development so we can get a really nice big root on this plant without suppressing mycorrhizal fungi. So mycorrhizal fungi are really important in the whole phosphorus acquisition for our plants. Effectively, what happens is the plant puts down their root, the mycorrhizal fungi will colonize that root and then explore further into the soil. So some people estimate it increases the effective rooting zone of the plant by 100 times or 1,000%, which is very important for phosphorus because it doesn't move at all in the soil. And so if we have a look over here, say this plant picks up all the phosphorus around its root zone, so within the half a centimeter around its roots, and it doesn't put any more roots out, there's no more phosphorus for it to absorb because it's only looking in the root zone. What mycorrhizal fungi do is it extends that root zone out 100 times, so making it really important or effective at finding new sources of phosphorus. So that's why phosphorus is really important, because it extends that root zone to access more area. It also assists with uptake of phosphorus. Now, the other biological component is phosphorus solubilizing bacteria. So there's things like Pseudomonas, Bacillus, Azotobacter, I think. You can see a list of them here. But basically, they're all these bacteria in the soil that help solubilize phosphorus. There's a few ways that these guys and the plant can uptake phosphorus. The first one is the plant obviously takes up the soluble phosphorus in the soil through the roots. So that's one way this takes up. But once this reserve is gone, the plant really needs help from the biology within the soil. So the first way, let's talk about organic phosphorus. So organic phosphorus is bound to carbon components, and there's really two ways that the plant can access these, all the microbes within it. They release an enzyme or two enzymes called phytase and phosphatase. These enzymes are released either by the plant itself, microbes or fungi, or the phosphorus solubilizing bacteria to decompose that organic phosphorus and releasing it to the plant as phosphorus. Now, the plant itself doesn't actually need to pick it up. The microbes or fungi can pick it up and transport it to the plant in exchange for carbohydrates or sugars that the plant produces through photosynthesis. So this is super important. But again, organic phosphorus can be broken up by phytase and phosphatase, as well as, I'm sure, different enzymes as well. But they're the two main ones, and I'm sure organic acids can help with that process. The next one is our absorbed phosphorus. Now for this one, the microbes or fungi, the roots, as well as the microbes, they release these organic acids to dissolve or break apart phosphorus from the clay or the aluminium ion oxides to release that into the soil solution for the microbes or fungi or the plant to then absorb. Now the important thing with this is that those organic acids can also chelate the phosphorus, meaning that it increases its or reduces its ability to be locked up again. So that can really increase the availability of the phosphorus. And then finally, we actually have the mineral component of phosphorus within our soil that the mycorrhizal fungi and the phosphorus solubilizing bacteria can access. And so this, again, with organic acids, they can break apart the phosphorus from the mineral component. Now, there's some really interesting studies about these tunnels that are drilled into the mineral component, and it's suggested that mycorrhizal fungi can actually do that. So because they're so small, they can get into these mineral components. They effectively drill into the minerals and access phosphorus that way to then supply it to the plant. Now, because of the size of the mycorrhizal fungi hyphae, it allows the mycorrhizal fungi to then access different nooks and crannies within the soil to a greater extent than our plant roots. So again, that's super important for accessing more phosphorus. Now, the final component I want to talk about is that the phosphorus solubilizing bacteria will associate with both the root through the rhizosphere, so that dreadlock around the root, as well as mycorrhizal fungi. So there's such bacteria that actually associate with the mycorrhizal fungi itself. So they're called mycorrhizal helper bacteria, and effectively they serve the same function that bacteria around the root would in terms of its relationship to the root, it would do for the mycorrhizal fungi hyphae. So you can think of this as more or less a highway or a network to exchange carbohydrates or carbon for phosphorus. So the plant produces the carbohydrates and the sugars, puts them into the roots, put them out to the mycorrhizal fungi. The mycorrhizal fungi then give them to the phosphorus solubilizing bacteria, and then up the chain it goes back to the plant. Super cool. So how do we actually make sure that we maintain a mycorrhizal fungi and a phosphorus solubilizing bacteria so that we can achieve this, so we don't have to apply as much fertilizer? Because remember, when we do the numbers, if we have to a meter's depth, which is not impossible for a plant to get down there, especially with mycorrhizal fungi, at 200 parts per million of phosphorus in the total component, which is more or less accessible. Not all of it is accessible, some of which is accessible to the plant or to the mycorrhizal fungi. Then we should have access to over 2,800 parts per million of phosphorus, or the equivalent to grow 800 tons of wheat. So there's actually a fair amount of phosphorus in that, and that's even for quite a poor or low phosphorus soil. So it's super important that we can rely on our mycorrhizal fungi to allow that to happen. So what can we do to achieve that? So the first component of that is, how do we increase our mycorrhizal fungi population? And the first place we can start about thinking of that is, how do we stop killing them? There's really a few ways to stop so that we can kill them. The first and the biggest one is tillage. A lot of Australian farmers are no-till, so more or less that's good. The other one are fungicides. Fungicides, obviously, mycorrhizal fungi is a fungi. Fungicides will hurt them, even if the label says it doesn't hurt them. It will definitely suppress them. Herbicides can suppress them. As I'm sure any other sides, especially nematicides. So really, any of those will hurt our biology, and it's not great. Now, a shallow tillage probably won't hurt them that much if they're in the subsoil, or it's especially below where it's getting plowed. It might even spread them, especially if you're putting a cover crop in straight after, or the crop straight after. But a lot of your herbicides will, especially if, now again, it's super difficult to get away from that. So not blaming anyone, just it's important to know that mycorrhizal fungi will react to these things. Now the other component is that the plants won't feed the mycorrhizal fungi if there's no reason for them to make that trade. Which means when we apply high rates of our fast releasing phosphorus, as well as high rates of our slow phosphorus, so both of these will suppress phosphorus in high enough amounts. Obviously, the fast releasing will release it all in one chunk, the slow at a time, but if there's enough of that, it's going to be great amounts as well. So from what I've seen, mycorrhizal fungi are suppressed once the phosphorus levels in our soil reach past that critical threshold. So what does that mean? It means that when we're making up our applications or determining our applications, we don't really want to go past that critical threshold on the soil tests. So the first point is we don't want to exceed our critical phosphorus value. So that's the first point. We want to get up to it, but we don't want to exceed it. Next, what we want to do is look at what we're applying. So it would be good to apply a combination of fast and slow so that we get both the root development that we want without suppressing. So we want to use both fast and slow phosphorus fertilizers. Now, a good place to start is potentially, especially if you're just starting with this, potentially 70% fast, 30% slow, and then eventually swap that over to 30% fast, 70% slow, and then completely to slow. That's one option in terms of spreading out that transition period. Doing that will assist with building up phosphorus solubilizing bacteria and potentially mycorrhizal fungi. Now, it's definitely possible to jump straight into this. If you're going to do that, I would recommend doing that on one paddock. So say 10% of your farm, you just jump straight into it. And on the other 90% of the farm, you just run it as usual. And then in 10 years time, you go across to all the paddocks, or you might just make sure that system works, and then you can do more and more and more. But overall, I'll do it slowly to make sure that this system actually works in your soils, because all these soils react so differently to phosphorus. And your farm would have a unique history with mycorrhizal fungi or lack of mycorrhizal fungi. Anyways, use fast and slow. If we're going to use the fast source, we want to basically convert that into a slow source. What we can do is add 5% humic acid to these applications. You can use humic acid or fulvic acid or humates. Any of the humic substances is fine, 5%. Humic acid tends to be a bit cheaper than fulvic. You can get soluble forms. One of our product suppliers has MAP that's coated in humic substances, so that's a really good one. What happens with this is that you add that humic or the carbon source to it, and what it does is it soaks up that phosphorus to prevent it from being fixed up straight away. So if you're going to do this, you buy your 5% humic substances, you apply it 5% to your product, and then you reduce your product application by the cost of your humic substance. So that's a really good way to balance out the costs. We're not reducing costs at this point, we're just making it more efficient to prevent suppressing our mycorrhizal fungi. So we want to make sure we build up to that the critical phosphorus component, but we don't want to exceed it, and then we want to go to a maintenance rate. When we go to the maintenance rate, what we want to do is we have our typical application, and we can start going back to somewhere between 5 to 10 kilos of phosphorus per hectare. So from what I've seen, this is kind of the Goldilocks zone with phosphorus application to develop root systems without suppressing mycorrhizal fungi. So that's somewhere, again, between 5 to 10 kilos of phosphorus, so that's straight phosphorus per hectare as our maintenance rate. Now, again, it's super important to take your soil tests into the future, because if this is not working, you need to adjust that and make sure it's actually working for your farm. So these are cool rules of thumbs, but you got to actually adapt this to your farm, which is why it's important to work with someone like us, where we can actually work with you to do that. So that works out to be somewhere between 20 to 40 kilos of MAP per hectare. So even with the 5% addition to our humic substances, of our humic substances to our applications, I wouldn't go below 5 kilos per hectare of phosphorus. With this, I would still include the humic substances at 5%, which is for, say, 20 kilos of MAP, that's going to be 1 kilo per hectare of the humic substances with the MAP. And this rate shouldn't suppress our mycorrhizal fungi. Now, again, don't jump straight to that because it might not work for your farm. What I would recommend is getting all your soil tests, making sure you know what's actually in your soil test. Do a typical year, including your 5% humic substances with what we talked about before with the typical application rates. Then take a differential sap test on your plants throughout the season. Not only that's going to assist with your current applications of foliars and, say, nitrogen management we talked about before, but it'll also help with, did this help with your phosphorus uptake? And what we can do over a few years is map that out as we slowly reduce our phosphorus applications and building up this side of things. So with that, with the differential sap tests, we know that phosphorus in the plant will move from the older leaves into the younger leaves. And basically we get this purple tinge coming out in the leaves when that happens. We can take a differential sap test, which will tell us that movement of those minerals within the plant. So we take an old leaf, we take a new leaf, send them to the lab, compare them, use our free differential sap test analysis tool, which looks like this. It'll tell you whether or not you have a deficiency in your plant. And then for phosphorus, now with the phosphorus, we kind of touched on the foliar applications of phosphorus aren't that efficient in season. It's more important that we get the starter right. If it's in season and we have a phosphorus deficiency, that informs us that what we did beforehand didn't work for the next season. I don't think you'll get any yield increases from a phosphorus application unless it's really bad, in which case I'm not too sure. You can trial it. The problem with phosphorus as a foliar is that it locks out with a lot of the trace minerals, and so really just making one application just for phosphorus, and it's a bit of a pain. But anyways, you can look at this from this season to inform next season's management. And if we can slowly step our way from typically what we're doing and slowly make our way through using differential sap tests to check what we're doing, I think that's a good way to approach it. So once we're at that point where we don't have excess phosphorus, we're just at that critical value or just below it, we're using fast and slow ferts, eventually just slow ferts. If we are using fast ferts, we're going to include humic acids, humic substances, and then we're not going to go past between 5 and 10% of kilos of phosphorus per hectare. Even with guano, we don't want to go too far outside of those. So all of that will stop suppressing our mycorrhizal fungi, which is great because we want to make sure we have the mycorrhizal fungi happening or the colonization happening at the same time. Because if we don't, and we just have fast fertilizer release, that will lock up after 6 weeks. And then what happens is that the plant hasn't formed any mycorrhizal fungi relationships, meaning that it will take another month or another 6 weeks to actually develop those symbiotic relationships if there's mycorrhizal fungi in the soil. So it can then take another 6 weeks of deficiency before it has access to more phosphorus. So that can be a problem. We want to make sure we supply it at the same time while developing our mycorrhizal fungi too. So we're going to do this, but we're also going to always, or maybe not always, but initially add a mycorrhizal fungi inoculant to our seed coats. This will cost you about $7 a hectare. So pretty cheap if basically you can cut all your phosphorus fertilizer down, and then you're just adding mycorrhizal fungi inoculants at $7 a hectare. Not bad. You can pretty much get away with that once with good, if you're going to maintain your mycorrhizal fungi populations. In the inoculant, you can also include our phosphorus solubilizing bacteria. Things like, you can actually get products designed for phosphorus solubilizing bacteria. You can have a look on our website for those, but also anything with a worm casting extract will have them in there, as well as things that fix nitrogen. So fantastic way to get both components on your seed and in your soil. So adding that to your seed coat, and then finally you can add those phosphorus solubilizing bacteria as a soil primer or compost or whatnot, so or amendment. So I like to do soil primers. I think they work out a bit cheaper and not as labor intensive as making your own compost. You add your soil primer, well, your molasses and your carbohydrates and your biostimulants, as well as your phosphorus solubilizing bacteria. The soil primer is really beneficial when you have that high amount of phosphorus build up in your totals. and so that will release into your available for next season. Now, in addition to this, what you can also do is add a cover crop. So the cover crop will basically do all of this for you, solubilize a bunch of phosphorus, lock it up as organic phosphorus in the roots of the system, as well as microbial biomass, as well as the litter. And then, so you got that in the organic component, and then your following crop can then release that more or less as a slow release fertilizer. So we can add that cover crop. Now there's a few species I would add to the cover crop to make it even more effective. I would add buckwheat, which buckwheat is really good at scavenging phosphorus. It releases a lot of phosphatase, so the stuff that breaks up the organic phosphorus. I'd add buckwheat. I think lupins also do it as well. So that's quite beneficial. Now, a lot of people hate on legumes. I don't think they're that bad. Including them can release a lot of acids into the soil, so be mindful of that. But the acids, depending on the acidity of your soil, can break apart the phosphorus binding to calcium. So if you're in quite basic soils, adding that could be really beneficial to bring your soil pH down, break up that phosphorus and that calcium, while also supplying nitrogen. Again, you might not want to do that if you don't need a lot more nitrogen, but who doesn't need more nitrogen? And if you're in highly acidic soils. So it kind of really just depends. But if you want a cover crop, what you can do is actually use our free cover crop mixer on our website. You get a custom cover crop mix. You can put in, we don't have a phosphorus solubilizing mix, but you can scavenge nutrients. So there's different species that help with that, as well as just overall soil health. The other thing with soil health and increasing the soil structure is that we can extend the root zone further into the soil as we improve it. So a plant that can only go, say, 15 centimeters deep into the soil profile won't have as much phosphorus available to it as a plant that can go half a meter. You've effectively more than doubled, tripled the soil area that you can mine for your plant. Now, there are a few concerns with what's called phosphorus stratification, which is typically seen in no-till systems where you're not inverting this topsoil down, and so you're not mixing up phosphorus, and phosphorus builds up in the topsoil, because if we're only placing fertilizer in the topsoil, then you can build up that. So there's a really interesting study looking at no-till, cover crops, the combination of the two, in terms of stratification of nutrients, especially phosphorus. What they found was super interesting. If you have cover crops by themselves, they create stratification. If you no-till by itself, it creates stratification. But if you actually use both of them, it reduces stratification. So the researchers don't understand why that happened or the mechanisms behind that. I'm not sure why, but it could have something to do with stimulating more microbes, fungi, or increasing biological activity to then share or cycle that phosphorus around. Likewise, earthworms can also move nutrients from topsoil into the subsoil and spread out that phosphorus too. Finally, there's another way you can actually increase or should be able to increase the amount of phosphorus taken up by your plants, and that's by increasing photosynthesis. The reason why is that the plant will increase its sugar production to then feed the microbes or fungi here, and the microbes or fungi will have more energy to go explore. So if we can increase the amount of root exudates to increase biological activity, then we should be able to increase phosphorus supply back up to the plant. The other component is that we can increase our soil organic carbon to hold more anions, so phosphorus, without them being locked up. So we can increase that, likewise with root exudates, to then hold more phosphorus in a more available form rather than it being locked up. So I think that's about it. Using this should be able to take your phosphorus applications down quite significantly without sacrificing yield. Now, I guess one of the worries is, are we mining our soils by doing this? And I guess so, potentially, because we're using the phosphorus already in our soils. I like to make sure there's good phosphorus in the topsoil because of that root development, but in the subsoil, there's a lot of phosphorus in that total component, and I think you almost have to mine it, otherwise you're bringing it in from other places that are effectively being mined. Phosphorus reserves in terms of the hard rock rock phosphate is being used up to make synthetics or inorganic fertilizers. So really, we're going to be mining phosphorus from somewhere. So they're going to be from our subsoil that has a bunch, or it's going to be from some mine somewhere. So there's really no sustainable phosphorus source because it's always going to be used. The only way that phosphorus can be really sustainable is if the product coming off your field then gets returned to your field. And the only way that can happen is what's called closing the poop loop, which is where human effluent then is returned back to the paddocks. That has a whole bunch of problems that I'm not going to get into today, and it's a part of a bigger picture in terms of management. The other component is that at the bottom of your soil profile, you have rock. Previously, higher up in your soil profile was that rock. So over time, you should be able to break up that rock with biological activity. I don't think we're there yet, and that might take thousands of years. The thing is, if the roots can go right down to your parent material and start breaking that up, there's a potential for that to release phosphorus as well. But that would only really come with extraordinarily high soil health, where you have biology touching your parent material. Don't know if that's possible. It's a potential to actually build up your soils like that. Awesome. Well, I hope you enjoyed the video. Make sure to reach out. We do offer free consultations for Australian farmers. If you want to get your foot in the door with regenerative agriculture, we'll give you 5 points that you can do to improve your operation today or this season. So reach out. We can help you with all of this. Thanks again for watching. My name's Teal. Cheers.